Hello, am I audible?
Yes, ma'am.
Okay, good evening, Alia. How are you?
Good evening, ma'am. I'm fine, alhamdulillah. How are you?
I'm good too. All right, so we were doing plant growth and development, right?
Yes, ma'am.
Okay. So do you have any doubts in differentiation, de-differentiation, and re-differentiation?
No, ma'am.
So that's clear, right? All right, now we'll talk about the developmental aspect of the chapter. Now, development is a term that includes all changes that an organism goes through during its life cycle from the germination of the seed to senescence. Everything that happens from that point is the developmental process. Now, the diagrammatic representation constitutes development of a cell of a higher plant. Now, see, this is how the development takes place. First of all, the meristematic cell is there, right? Now, meristematic cell actually grows in terms of its size by plasmatic growth, meaning the plasma grows, meaning the cell size grows. Then by de-differentiating, it turns into a mature cell. Now this mature cell, after like once it has served its purpose, it undergoes the period of senescence, that is old age in plants, and then ultimately leading to the death. But this mature cell can also go back to the normal state. We know that by studying the differentiation and redifferentiation. Now, plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability of plants to actually see, depending upon what environment condition that they prefer and what is their, you can say, condition at that point of time, they can actually change the way they look, and that is plasticity. One example, or one very common example of plasticity is heterophyll in cotton, coriander, and larkspur. Hetero means different, right? Multiple and different kinds. And phylly is for the phenomena, because we know that phyllotaxy and phylly is for the leaves. So heterophylly is different kinds of leaves. So in such plants, the leaves of the juvenile plants are different in shape from those in mature plants. The same plant will have different kind of leaf when younger, and when being an adult, they would become different. On the other hand, difference in shapes of leaves produced in air and those produced in water in buttercup also represent the heterophilous development due to environment. This phenomena of heterophily is an example of plasticity. So always remember that example of plasticity, meaning the way the plant responds is heterophily. This is these are the pictures. Now, and see one more thing, like the same for like buttercup, the buttercup leaves on a terrestrial habitat are like this. But on a water, in a water habitat, it will become like this. So depending upon the situation and depending upon the environment, the plants can adapt themselves or have different kinds of, you can say, structures in the body, and that is plasticity. Now moving forward, thus growth, differentiation, and development are very closely related events in the life of a plant. Broadly, development is considered as the sum of growth and differentiation. Development in plants, that is both growth and differentiation, is under control of intrinsic, that are internal, and extrinsic, that are external factors. Now, the former one, meaning the intrinsic, that is the internal factor, it includes the intracellular, that is within the cell, that is the genetic, or intercellular, that is in between the two cells, factors such as the plant growth regulators or the plant hormones. Now, while the latter includes light, temperature, water, oxygen, and nutrition, this is for extrinsic. Extrinsic means the external factors that affect the plant's development. Now, talking about the plant growth regulators. The plant growth regulators, or PGR, are small, simple molecules of diverse chemical composition. They could be indole compounds, meaning derived from indole, such as the indole acetic acid, that is auxin. They are derived from the indole compound. Now talking about the adenine derivative. Adenine, they have been derived from the adenine, that is furfuryl aminopurine, that is kinetic. Then there are derivatives of carotenoid, which is a pigment. Then there is abscisic acid or ABA. The derivatives of terpenes, that is gibberellic acid, and the derivatives of gases such as ethylene. So here the source of these hormones are very important to remember, because many a times they are asked in the question, like what are the sources of the hormones, like what kind of biomolecule has the hormone been derived. So that is very important to remember. Now, the PGR can be broadly grouped into two groups based on the functions in the living plant body. One of the PGRs are involved in growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. All the growth in terms of positive aspects from the plant, that is the growth regulators or growth promoters. These are also known as plant growth promoters. Now, the examples are auxin, gibberellin, and cytokine. The PGRs of other group play an important role in the plant responses to wounds and stresses of biotic and abiotic origin. So, the ones that the other group of hormones that act or they perform when there is a stressful situation for the plant, also when the plant gets injured, or in the case of senescence, when the plant now is undergoing a period of, you can say, old age. So at that time, a different set of hormones, PGRs, would act, and those are the plant growth inhibitors. And the best example for that is the abscisic acid. They are the ones that inhibit the growth activity. Clear? Any doubts up till now?
Yes, ma'am.
No.
No, ma'am.
Yes, okay. They are also involved in various growth inhibiting activities such as dormancy and abscission. The PGR abscisic acid belongs to this group. The gaseous PGR, that is the ethylene, could fit in either of the group. Now, depending upon the situation, the ethylene, which is a gaseous hormone of the plants, it can act like a growth promoter also. It can act like a growth inhibitor as well. But mostly it is an inhibitor. Now talking about the discovery of the plant growth regulator. This one is the most important. This coleoptile example. The discovery of each major groups of PGR have been accidental. All this started with the observation of Charles Darwin and his son Francis Darwin, when they observed that the coleoptiles of the canary grass responded to the unilateral illumination by growing towards the light. For example, if this is the grass, and now when the light was provided to this from one side, the grass actually bent towards the light. So they studied this thing. This mechanism is actually known as phototropism. Now, after a series of experiments, it was concluded that the tip of the coleoptile was the site of transmittable influence that caused the bending of the entire coleoptile. Auxin was isolated by F.W. Wendt from the tips of coleoptile of oat seedlings. Now, auxin was actually isolated from oat seedling, but it was discovered for canary grass. What happens is that whenever, let's see, auxin is predominantly made and acts at the tip of the grasses or any plant. And what it does is that, for example, light is coming from this end towards here. Now, auxin will shift to the one side that does not get the light, and it starts the cell division process, because of which the tip slowly bends towards the light. Now, the Bacani, that is foolish seedling disease of rice seedlings, was caused by a fungal pathogen that was Gibberella fujikuri. Now, a person who was E. Kurosawa in the year 1926 reported the appearance of symptoms of the disease in rice seedlings when they were treated with sterile filtrates of the fungus. The active substances were later identified as gibberellic acid. So gibberellic acid causes a lot of differentiation, and that is how it was extracted and found out. Now, F. Scoop and his co-workers observed that from the internodal segments of tobacco stem, the callus, which is a mass of undifferentiated cells, proliferated only if, in addition to auxins, the nutrient medium was supplemented with one of the following, that is extract of the vascular tissue, yeast extract, coconut milk, or DNA. When auxin was, combined with these extracts, then they show a lot of division activity. So Miller et al., Miller along with all his colleagues, later identified and crystallized the cytokinesis promoting active substance that they termed as kinin. So by this, cytokinin was extracted and observed. During mid-1960s, three independent researchers reported the purification and chemical characterization of three different kinds of inhibitors. Inhibitor B, abscission 2, and dormant. Three kinds of inhibitors that are there. Later, all the three were proved to be chemically identical. It was named abscisic acid. So whether it is inhibitor B, abscisic 2, or dormant, all of them causes the inhibition of the plant's growth. So they were altogether, they were same only. Like their chemical composition were identical to each other. Because of this, they were kept and they were named as ABA, that is abscisic acid. Now, H. Cousins in the year 1910 confirmed the release of a volatile substance from ripened oranges that hastened the ripening of stored unripe bananas. Later, this volatile substance was identified as ethylene, which is a gaseous plant growth regulator. Now, we'll talk about each of their physiological effects that they have on the plant. Talking about the auxin. Auxins, the name auxin has been derived from the Greek word that is auxein, means to grow. It was first isolated from human urine. The term auxin is applied to indole 3-acetic acid. Auxin is the common name, indole 3-acetic acid is its chemical name, and to other natural and synthetic compounds having a certain growth regulating properties. They are generally produced by the growing apices of the stem and the root. So it is present at the apex, from where they migrate to the regions of their action. Auxin, like indole acetic acid and indole butyric acid, IBA, have been isolated from plants. Naphthalene acetic acid, that is NAA, and 2,4-D, that is 2,4-dichlorophenoxyacetic acid, that these are the synthetic auxin. This is important. NAA and 2,4-D are synthetic auxin, meaning they are chemically prepared by us. All these auxins have been used extensively in agriculture and horticulture practices. Now, what are the functions? They help to initiate rooting in stem cutting. They cause rooting, formation of roots. An application widely used for plant propagation. Auxins promote flowering, example in the case of pineapple. They help to prevent fruit and leaf drop at early stages, but promote the abscission of older mature leaves and fruits. When the leaves and fruits have become mature, now they are like no more in need of nutrients. So auxin will cause them to drop or cause the abscission. Now in most higher plants, the growing apical bud inhibits the growth of the lateral buds, a phenomenon known as apical dominance. Removal of the shoot tips, if we cut off the shoot tips, so this usually results in the growth of lateral buds. It is widely used in application for the tea plantation, hedge making. The apical dominance is when the apical bud does not let the lateral buds to grow. Because of this, the shoot tips are cut down so that the lateral buds would go, because if the apical bud is not there, then the auxin will not be there for division constantly. So the lateral buds will grow. Auxin also induce parthenocarpy, example, tomatoes. They are widely used as herbicides to kill the herb, like the weeds and everything. And 2,4-D is widely used to kill dicotyledonous weed. Does not affect mature monocot plants. It is used to prepare weed-free lawns by gardens. Auxin also controls xylem differentiation and help in cell division. So all the positive aspect of the growth in the plants, that is actually because of the auxin. Now talking about gibberellins. Now gibberellins are another kind of promotory PGR. There are more than 100 gibberellins reported from widely different organisms such as fungi and higher plants. They are denoted as GA1, GA2 and GA3 and so on. However, gibberellic acid GA3 was one of the first gibberellins to be discovered and remains the most intensively studied form. This is the one that actually we study also, and this is the one which acts on it the most. Okay. All gibberellic acids are acidic. They produce a wide range of physiological responses in the plants. Their ability to cause an increase in length of axis is used to increase the length of grape stalks. Gibberellins cause fruits like apple to elongate and improve its shape. They also delay senescence, meaning they will not allow the old age or the, you can say, negative aspect of growth to come to the plant. Thus, the fruits can be left on tree longer so as to extend the market period. GA3, gibberellic acid 3, is used to speed up the malting process in the brewing industry. So it is also used in malting processes. Sugarcane stores carbohydrate as sugar in their stems. Spraying sugarcane crop with gibberellin increases the length of the stem, thus increasing the yield by as much as 20 tons per acre. So when the length of the stem is increased, so as the sugar quantity in it. Spraying juvenile conifers with GAs hastens the maturity period, thus leading to early seed production. Gibberellins also promote bolting, that is internode elongation just prior to flowering in beet, cabbages, and many plants with rosette habitat. Okay, so such as in cabbages like that have a flower-like habitat, they help in the internode elongation so that there will be like proper spacing in between each of the rosette. Any doubts up until now?
No,
Any
ma'am.
doubt? Okay. Now talking about the cytokines. Now cytokinins have specific effects on cytokinesis and were discovered as a kinetin, that is a modified form of adenine, which is a purine, like a purine. So you must remember where like each of the hormones, where they are derived from is important. Now from the autoclaved herring sperm DNA. Kinetin does not occur naturally in plants. Search for natural substances with cytokinin-like activities led to the isolation of zeatin from the corn kernels and coconut milk. Since the discovery of zeatin, several naturally occurring cytokines and some synthetic compounds with cell division promoting activity have been identified. So initially, the naturally occurring cytokinins were not found, but later on it had been identified. Natural cytokinins are synthesized in the regions where rapid cell division occurs. For example, the root apices, developing shoot buds, young fruits, etc. It helps to produce new leaves, chloroplast in leaves, lateral shoot growth, and adventitious shoot formation. Cytokinins help overcome the apical dominance. They promote nutrient mobilization, which helps in the delay of senescence. Again, this is also delaying the senescence aspect. Now, talking about the ethylene. Ethylene is a simple gaseous plant growth regulator. It is synthesized in large amounts by tissues undergoing senescence and ripening fruits. Influences of ethylene on plants include horizontal growth in seedlings, swelling of the axis, and apical hook formation in dicot seedlings. Ethylene promotes senescence and abscission of plant organs, especially of leaves and flowers. Ethylene is highly effective in fruit ripening. What happens is that, for example, if there is a fruit and it is starting to ripen. Now, if beside that fruit we keep an unripened fruit, the gaseous EPGR, that is ethylene, which is released by the ripening fruit, will cause the ripening of the unripe. It enhances the respiration rate during ripening of the fruits. This rise in the rate of respiration is known as respiratory climactic. Again, important. Ethylene breaks seed and bud dormancy, initiates germination in peanut seeds, sprouting of potato tubers. Ethylene promotes rapid internode or petiole elongation in deep water rice pods. It helps leaves or upper parts of the shoot to remain above the water. Ethylene also promotes the root growth and root hair formation, thus helping the plants to increase their absorption surface. These are the functions of ethylene. Now, why and where do we apply it? So, it is used to initiate flowering and for synchronizing fruit set in pineapples. It also induces flowering in mangoes. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture. So again, a question may be asked as to which is the most widely used plant growth regulator. Now, the most widely used compound as a source of ethylene is ethifon. Ethifon, you can consider it as the artificial, so you can say synthetic ethylene. Now, ethifon is an aqueous solution. It is readily absorbed and transported within the plant and releases ethylene slowly. Ethifon hastens fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits, that is thinning of cotton, thinning of cherry, walnut. It promotes female flowers in cucumbers, thereby increases the yield. These are all like ethylene has a lot of functions to perform and it is actually applied in multiple settings. Why? Because it is very easy to use. It has the most effective power as compared to the other hormone. Now talking about the abscisic acid. Now this is a plant growth inhibitor. It regulates abscission and dormancy. But like other PGR, it also has other wide-ranging effects on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of plant metabolism. Abscisic acid inhibits seed germination. ABA stimulates the closure of stomata and increases the tolerance of plants to various kinds of stresses. Now, see, closure of stomata, why is it doing so? That not excessive water loss can take place from the plant's body. So, this is the reason why it is known as a stress hormone. Abscisic acid plays an important role in seed development, maturation, and dormancy. By inducing dormancy, abscisic acid helps seeds to withstand desiccation and other factors unfavorable for growth. In most situations, the abscisic acid acts as an antagonistic to gibberellic acid, meaning antagonistic means it will act the opposite or against the gibberellic acid. So we may summarize that for any and every phase of growth, differentiation and development, one or the other PGR has some role to play. Such role could be complementary or it could be antagonistic. These could be individualistic or synergistic. Individualistic, that one hormone is acting alone. Synergistic is a combined effect of more than one hormone. Similarly, there are a number of events in the life of a plant where more than one PGR interact to affect that event, such as dormancy in the seed or dormancy in the bud, the abscission, senescence, and apical dominance. Here, more than one PGR would actually act on it. For example, if we're talking about dormancy in seed, so it can be a collective effect of abscisic acid and ethylene. Remember, the role of PGR is only one kind of intrinsic control. Along with genomic control and extrinsic factors, they play an important role in the plant growth and development. Many of the extrinsic factors, such as temperature and light, control plant growth and development via PGR. So all it is a cumulative effect, meaning the PGR will not only work if the extrinsic factors such as the temperature, pH, light are not there. Some of the events could be vernalization, flowering, dormancy, seed germination, and plant movement. Now, this thing, vernalization and photoperiodism has been removed from your syllabus, right?
Yes, ma'am.
Okay. So with this, we have completed the chapter that is plant growth and development. Now you tell me if you have any doubts anywhere. Did you understand the topic?
Yes, ma'am.
All right. Okay, so now you take 5 minutes, go through the chapter once, see the topics once, and tell me if you have any doubts. And if you don't, then we'll start a new chapter.
Okay, ma'am.
Yes, dear, any doubts?
No, ma'am.
Whole chapter is clear, right?
Yes, ma'am.
Okay, so now we are going to start with the next chapter, probably the last chapter, right? It's the last chapter for class 11. That is biomolecules, isn't it? This is the only chapter that was left, right?
Yes, ma'am.
Yeah, rest all the chapters for 11th class, like it's done. Okay. Now talking about biomolecules, these are the substances that are present inside our body also, in the elements also, in the whole entire world. Now, the biomolecules can be divided into biomacromolecules and biomicromolecules. These are the two types that we can divide them. Now, okay. Now, if we do a chemical analysis as to what are the components for plants and animal tissue, we'll find like majority of things coming out of them. So, if we perform such an analysis on plant tissue, animal tissue, or on a microbial paste, we obtain a list of elements such as carbon, hydrogen, oxygen, and several others, and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of Earth's crust as an example of non-living matter, we obtain the similar list because it's there on the Earth's crust as well. Now, what are the differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in the sample of Earth's crust are also present in the sample of the living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in Earth's crust. Now, that's true that we may not find selenium or mercury inside our body, but carbon and hydrogen is maximum because we are made up of that. Now, how to analyze chemical composition? We can continue asking in the same way what type of organic compounds are found in living organisms. How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue that is either a vegetable or a piece of a liver for animal tissue and grind it in trichloroacetic acid using a mortar and pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or a muslin cloth or a cotton, we would obtain two fractions. The one is the filtrate, or more technically, the acid soluble pool, and the second is the retentate, that is the acid insoluble fraction of it. Scientists have found thousands of organic compounds in the acid soluble pool. Okay. All carbon compounds that we get from the living tissues can be known as the biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One weighs a small amount of living tissue, say a leaf or liver, and this is called the wet weight. And then we dry it. All the water evaporates, then we'll measure the dry weight. Now, if this tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, that is CO2, water vapor, and are removed. Now, what is remaining is the ash. Now, this ash contains inorganic elements such as calcium, magnesium, inorganic compounds such as sulfate, phosphate, which are also there in our body. Therefore, elemental analysis gives elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compounds gives an idea of the kind of organic and inorganic constituents. So, these are the list. For example, if you're talking about the elements that are present in the non-living and the living matter, the percentage of each of those has been given here. and also about the inorganic constituents that are found in the living tissue. Sodium, potassium, calcium, magnesium, water, and various compounds are also there. Now, from a chemistry point of view, one can identify functional groups like aldehyde, ketones, aromatic compounds, etc. But from a biological point of view, we shall classify them into amino acid, nucleotide bases, and fatty acids. Now, amino acids are organic compounds containing an amino group, that is NH2 group, and an acidic group as substituents on the same carbon, that is the alpha carbon. So one carbon will have two groups attached to it, or two functional groups. One will be the NH2 group and one will be the acidic group, that is most probably the carboxylic acid. So these types of amino acid that have both the groups attached onto them, these are the alpha amino acids. Now they are substituted methanes. Therefore, there are four substituent groups occupying the 4 valency position: hydrogen, carboxyl group, amino group, and a variable group. We know that carbon can form 4 bonds, right? So each of those 4 bonds will be the 4 functional group attached. Based on the nature of the R group, there are many amino acids. R group is the variable group. However, those which occur in proteins are of only 20 types. One protein is made up of 20 amino acids. Now, the R group or the variable group in these proteinaceous amino acid could be a hydrogen. So, if the R group is a hydrogen, then that amino acid is glycine. If it is a methyl group, then that is alanine. If it is hydroxyl methyl, then that is serine. So 3 of the 20 are shown in the figure 9.1. See, where is it? Where is it? Oh, here. See this. See, all of them have same compositions as to acid, acid, acid. Then amine group, amine group, amine group. One will be hydrogen, hydrogen, hydrogen. So 3 groups are fixed. Now the differences between each of the amino acid is because of the R group. That is the variable group. So it can be hydroxyl methyl and become serine. If the R group is a methyl group, that is alanine, and if the R group is hydrogen, then that is glycine. The chemical and physical properties of amino acids are essentially of the amino, carboxyl, and the functional group. Based on the number of amino and carboxyl groups, there are acidic. Now, if any amino acid has more acid component to it, then that becomes a acidic amino acid, such as the glutamic acid, aspartic acid. If it has more NH2 group, then that becomes a basic amino acid, such as lysine. If it has both in equal number, then that becomes a neutral amino acid, that is valine. Similarly, there are aromatic amino acids such as tyrosine, phenylalanine, tryptophan. A particular property of amino acid is the ionizable nature of NH2 and COOH groups, meaning it can exist like a negatively charged molecule as well as a positively charged molecule all together in one, and that is a Zwitter ionic form, and this is the most, you can say, unique property about them. Now, lipids are generally water insoluble. They cannot, now we're talking about lipids now. So they are water insoluble. They could be simple fatty acid. A fatty acid has a carboxyl group attached to an R group. The R group could be methyl or ethyl, or higher number of the CH group, such as 1 to 19 carbons, up till 19 carbons. For example, palmitic acid has 16 carbons, including the carboxyl carbon. Arachidonic acid has 20 carbons, including the carboxyl carbon. Fatty acid should be or could be saturated, meaning without double bond, or it could be unsaturated, that is with double bonds, more than one, like double bonds. Another simple lipid is glycerol, which is trihydroxypropane. This is the chemical name for the glycerol. Many lipids have both glycerol and fatty acids. Here, the fatty acids are found esterified with glycerol. It forms an ester with the glycerol. They can be monoglyceride, diglyceride, and triglyceride. These are also called as fats and oils based upon the melting points. We know that oils are in a liquidy form, so they have lower melting points, such as gingerly oil, and hence remain as oil in winters. Now, some lipids have phosphorus and a phosphorylated organic compound in them, so we'll call them as phospholipid. One such example which is found in our body is found in the cell membrane, that is lecithin. Some tissues, especially the neural tissues, have lipids with more complex structures because they have a proper layering, and the neural tissues are surrounded by the lipid molecules for insulation purpose as well. Now, living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogenous bases. Now, nitrogenous bases are of two types, that is purine and pyrene. Now, one like the names of the nitrogenous bases are adenine, guanine, cytosine, uracil, and thymine. When found attached, now when nitrogenous base forms a bond with the sugar, that is nucleoside. And if a phosphate is added to a nucleoside, then that becomes a nucleotide. Now, adenine, guanosine, thymidine, pyridine, cytidine are nucleosides, meaning nitrogenous base, for example, adenine plus sugar is equal to adenosine. Guanine plus sugar is equal to guanosine. Cytosine plus sugar is equal to cytidine. But when they are attached with the phosphate group, for example, adenosine plus phosphate group becomes adenylic acid. Guanosine plus phosphate group becomes guanylic acid. So depending upon the groups that are they attached to, they can be nucleoside and nucleotide. Nucleic acids such as DNA and RNA consist of nucleotides only. DNA and RNA function as genetic material. Now we know that the ribonucleic acid, both of them are the genetic material, right? So they are the genetic material, and remember that the nucleic acids are polymers of nucleotide, meaning they are made up of several nucleotides. Any doubts up until now?
No, ma'am.
Okay. Now talking about the primary and secondary metabolites. The most exciting aspect of chemistry deals with the isolating thousands of compounds, small and big, from living organisms, determining their structure and, if possible, synthesizing them. If one were to make a list of biomolecules, such a list would have thousands of organic compounds, including amino acids, sugars, etc. For reasons that are given, we can call these biomolecules as metabolites. In animal tissues, one notices the presence of all such categories of compounds. See, metabolites are those compounds that are found in the body of the living organism, whether it is a plant or whether it is an animal. Now, the ones that are involved in the structural and the body aspect of the organism, they are the primary metabolites, meaning because of this, the organism cannot exist. Whether it is a plant or an animal, it doesn't matter. Now, talking about the plants, now plants have the ability to make certain extra types of metabolites that they do so for their defense. Okay, so those metabolites that are not involved in the body structure and the living of the plants, then that is the secondary metabolite. This is the various components that are of use to humans, but are of no use to them. Now, when one analyzes plant, fungal, and microbial cells, one would see thousands of compounds other than these called primary metabolites, meaning other than the primary metabolites such as alkaloid, flavonoid, rubber, essential oil, antibiotics, colored pigments, scents, gums, and spices. These are known as secondary metabolites. While primary metabolites have identifiable functions and play known roles of normal physiological process, meaning growth, reproduction, and everything. Secondary metabolites are of no use to the plants. Rather, it is useful for us humans. We use rubber for making like paper and everything. Latex is used for making paper, which is again of no use to the plant. Then many secondary metabolites that are found in the plant's body are used for making medicines for humans. Then spices, we use that in cooking. Scents, we apply it to like the fragrances that are available from the plants. They are of no use to the plants, but they are of use to us. And then again, pigments. Now this chart is very important, very important. They can ask you like the example of pigments that are found, the alkaloids. So these examples are very important to remember. The names of the toxins such as abrin, ricin. Then what are the essential oils such as lemongrass essential oil. Then there is lavender essential oil. So the essential oils are also extracted from the plant only. Now some secondary metabolites have ecological importance because it is for us, right? Spices are such a like great trade and economy related aspects that are obtained from the plants. We earn money because of that. Now we'll talk about biomacromolecule, and this chart is very important. So you learn it, yeah, learn it by heart. Now talking about biomacromolecules. There is one feature common to all those compounds found in the acid-soluble food. They have molecular weights ranging from 18 to 800 Dalton approximately, meaning they have high molecular weight. And they are found in the acid-soluble food. What are the biomolecules? Biomacromolecules such as proteins, nucleic acid, lipids, and polysaccharides. Now, acid insoluble fraction, talking about the ones that do not solubilize in the acid. It has four types of organic compounds: proteins, nucleic acid, polysaccharides, and lipids. These classes of compounds, with the exception of lipids, have molecular weight in the range of 10,000 Daltons and above. So they have higher, you can say, molecular weight, except for lipids. For this very reason, the biomolecules, that is, chemical compounds found in the living organism, are of two types. Ones that have less than 1000 Dalton, and they are referred to as micromolecules, while the other ones that are greater than the 1000, they are the biomacromolecules. Now, the molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Polymeric substances are those that are made up of several monomers. Then why do lipids, despite having a smaller size, meaning their molecular weight is not more than 800 Dalton, but still they come under the acid insoluble fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles and are not water soluble. So since the lipid comes in the acid insoluble food, that is why it has been kept in the biomacromolecule. But again, remember that they are not strictly macromolecule because their size or their molecular weight is very small. Now, the acid soluble pool represents roughly the cytoplasmic composition. Macromolecules from the cytoplasm and organelles become the acid insoluble fraction. So, now again, the components of the living cell. This is also very important. Like what percentage of what component are found? Like water, 70 to 90% of the cell. Proteins, 10 to 15. Carbohydrates, 3. Lipids, 2. Nucleic acid, 5 to 7. And ions, 1. So these are all the components. Now talking about the most important biomacromolecule, that is protein. Now protein, they are polypeptides. Proteins are made up of amino acids. One protein is made up of 20 amino acids. They are linear chain of amino acid. Linear means in a line-like fashion. And they are linked by peptide bond. Each protein is a polymer of amino acid, as there are 20 types of amino acids. Example, alanine, cysteine, proline, tryptophan, lysine, phenylalanine. So many amino acids are there. A protein is a heteropolymer. Heteropolymer means that it is made up of different kinds of amino acids, not the same kind of amino acid. Now, a homopolymer has only one type of monomer repeating n number of times. This information about the amino acid content is important, as in later you will learn that they are very essential for health. Now, the amino acid can be essential or non-essential for us. Those that are essential, or you can say first let's talk about non-essential ones. Non-essential ones are the ones that we take through our diet. But the essential ones are something that we are not able to take through our diet, so we have to take it externally. Proteins carry out many functions in the living organism. Some transport nutrient across the cell, some fight infectious agents, and some are the hormones. Now again, this chart is so important. It is very important as to telling the functions of the protein, like the functions of each different kinds of proteins, such as GLUT4 is for transportation of glucose. This came in NEET 1 year. And then receptor, the protein also acts as receptor. The protein acts as hormones. So proteins are also enzymes, right? Many enzymes are proteinaceous in nature. So proteins are very important biomolecules. Now, amongst that, collagen is the most abundant protein in animal world. And we have studied photosynthesis, so we know that Rubisco enzyme, which is a proteinaceous enzyme, it is the most abundant protein in the whole biosphere. So this was about protein. Any doubts?
Ma'am, your voice was cracking at the end. Can you repeat what you just said?
Yeah, I said that the collagen, like this, first of all, this chart is very important as to what the protein is, like the different kinds of proteins and their functions. Apart from that, we have learned that the collagen is the most abundant protein for animal world. Okay, because it is found in the skin. But talking about Rubisco, we have studied this in photosynthesis, that Rubisco is the most abundant protein in the world. And this is an enzyme, right? Where is it used? Rubisco is used where? Hmm? Rubisco is used where? It was there in the photosynthesis chapter, right?
Yes, ma'am.
Yes. So where is it used? Tell me.
Calvin cycle,
Yeah. And what does it do actually?
carboxylation rate.
Hmm. Now talking about polysaccharides. Now the acid insoluble pellet also has polysaccharides. Now the monomers of polysaccharides are several. What? Monosaccharides. Now polysaccharides are long chains of sugars. They are threads, literally a cotton thread containing different monosaccharide as building blocks. For example, cellulose. It is a polymeric polysaccharide, consists of one type of monosaccharide, that is glucose. So cellulose is a polymer of glucose, and cellulose is a homopolymer, meaning only glucose so makes the cellulose, meaning several glucose molecule makes up the cellulose. Starch is a variant of this, but present as a storehouse of energy in plant tissues. It is the stored house, or you can say reserve food material for plants. Now starch is also a polymer. Now animals have another variant called glycogen. This is again a polymer of glucose. Now, inulin, it is a polymer of fructose. In polysaccharide chain, say glycogen, the right end is called as the reducing end and the left end is called as the non-reducing end. It has branches as shown in the form of cartoon here, this one. The right end and the left end, both of them have functions. The right end is the reducing end and the left end is the non-reducing end. It has branches. Now, starch forms helical structures, meaning in their secondary state, the starch will form helix-like structure. In fact, starch can hold iodine molecules in the helical portion. The starch iodine is blue in color. Cellulose does not contain complex helices and cannot hold I2, that is iodine. So starch can hold iodine. That is why whenever we want to check whether the plant is doing photosynthesis or not, we do iodine test on it. So if the plant that does photosynthesis, if it turns blue, it means that it has starch in it and starch is the end product, right? So that is why we do iodine test to observe whether photosynthesis is done or not. Plant cell wall are made up of cellulose. Paper made from plant pulp and cotton fiber is also cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino sugars and chemically modified sugars, that is glucosamine and acetyl galactosamine, NAG and NAG. Okay, now exoskeletons of arthropods, for example, have a complex polysaccharide that is chitin. These complex polysaccharides are mostly homopolymers. Most of the polysaccharides are homopolymers. Now, these are like every single example of polysaccharide, pay attention to that as well. Now, talking about the nucleic acid, the other type of macromolecule that one find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with polysaccharides and polypeptides, these comprise the true macromolecule fraction of any living tissue or cell. For nucleic acid, the building block is a nucleotide. A nucleotide has three compounds. We have the heterocyclic group, that is the either a purine or a nitrogenous base is there. Then a sugar is added and then a phosphorate or phosphoric acid is added or phosphate is added. The heterocyclic compounds in nucleic acid are the nitrogenous base, adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted purines. See, nitrogenous bases are of two types. We have purines and we have pyrimidines. Now, under the purines, we have adenine and guanine, and under the pyrimidines, we have what? Cytosine, thymine, and uracil. The skeletal heterocyclic ring is known as a purine and pyrimidine respectively. The sugar found in the polynucleotide is either ribose sugar, that is a monosaccharide pentose. Pentose means 5-carbon sugar. Or it can be deoxyribose sugar. A nucleic acid containing deoxyribose is known as deoxyribonucleic acid, that is DNA, while that contains ribose is the RNA, ribonucleic acid. Now talking about the structure of protein. Protein, as mentioned earlier, these are the heteropolymers that contain the strings of amino acids. Structure of molecules mean different things in different contexts. In inorganic chemistry, the structure invariably refers to the molecular formula. Organic chemists always write a 2D view of the molecule while representing the structure of the molecule, such as benzene, naphthalene, etc. So now we'll also talk about the structure of proteins, how it looks like in different stages. Now, the sequence of amino acid, the positional information in a protein, which is the first amino acid, and then which is the second, we'll talk about in that way. The first structure of the protein is the primary structure. A protein is imagined as a line, the left end represented by the first amino acid and the right end represents the last amino acid. So linear structure is a protein's primary structure. Now the first amino acid is also known as the N-terminal amino acid. The last amino acid is called as the C-terminal amino acid. A protein thread does not exist throughout as an extended rigid rod. The thread is folded in the form of a helix. So when it gets folded in the form of a helix, or it gets in the sheeted form, so then that becomes a secondary structure. The thread is folded in the form of helix. Of course, only some portions of the protein thread are arranged in the form of helix. In proteins, only right-handed helix are there. Other regions of the protein thread are folded into other forms, and this is how we call as the secondary structure, meaning when the linear structure starts getting helical or sheeted, then that becomes a secondary structure. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, just like how the proteins exist. Okay, so this is the tertiary structure. Now, this is the most important structure in the field of biology for any working of proteinaceous compound. This is a 3D view of a protein. Tertiary structure is absolutely necessary for the many biological activities of protein. Also, the tertiary structure is the structure which is found in the enzymes. Now, lastly, there is the quaternary structure. Here, some proteins are assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other, and then it forms a proper, like, see, a quaternary structure is the, you can say, the largest structure of the protein, and it has 4 subunits. Quaternary, it is the 4th level. Linear string of spheres arranged one upon each other in the form of cube or plate is the architecture of a protein, otherwise called as the quaternary structure of a protein. Adult human hemoglobin consists of 4 subunits. 2 of these are identical to each other, hence 2 subunits are alpha type and 2 subunits are beta type, together constituting the human hemoglobin. Now, human hemoglobin is quaternary structure. Enzymes are tertiary structure. Now, talking about the enzymes. Almost all enzymes are proteins. There are some nucleic acid that behave like enzymes. These are known as ribozymes. One can depict an enzyme by a line diagram. An enzyme, like any protein, has a primary structure, that is amino acid sequence of the protein. An enzyme, like any protein structure, has the secondary and the tertiary structure. When you look at a tertiary structure, you will notice that it has folds upon itself. The chain crisscrosses itself, and hence many crevices or pockets are made. If we make things like this, there are so many ways that curvings are there, right? These are the pockets or the crevices. One such pocket of the tertiary structure is the active site. An active site of an enzyme is a crevice or a pocket into which the substrate fits. The substrate is a compound on which the enzyme acts upon. Thus, enzymes, through their active site, catalyze reactions at a high rate. Enzyme catalysts differ from inorganic catalysts in many ways, but one major difference needs mention. Inorganic catalysts work efficiently at high temperatures and high pressure, while enzymes get damaged at high temperature, that is above 40 degrees Celsius. However, enzymes that are isolated from organisms who normally live under extremely high temperatures, such as hot vents, sulfur springs, they are stable. Meaning, for those organisms that already live in a very high temperature environment, they have the enzymes that work at high temperature, that is 80 to 90 degrees Celsius. But normally the enzyme can get denatured above the 40 degrees Celsius. And the way by which the enzyme's tertiary structure gets destroyed, that is known as denaturing. And it occurs at temperatures above 40 degrees Celsius. Thermal stability is thus an important quality of such enzymes isolated from thermophilic organisms. They are thermal stable, meaning they can be stable. The enzyme structure won't get destroyed at higher temperature. Now talking about chemical reactions, how do we understand these enzymes? Let us first understand a chemical reaction. A chemical compound undergoes two types of changes. There is physical change, simply refers to a change in shape without the breaking of bond. Here, no new substance is formed. Another physical process is a change in state of matter. For example, when ice melts into water, from solid it's getting into liquid, or when water becomes a vapor from liquid to gas. These are physical processes and they are reversible. But when the bonds are broken and new bonds are formed, new molecules are formed, then that is a chemical reaction. For example, any of the reaction that occurs in biology. Now, this is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Inorganic means outside the body that occurs in the environment, and organic is the one that occurs inside the body. The rate of a physical or a chemical process refers to the amount of product formed per unit time. The rate is the product formed per unit time. Rate can also be called as the velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperature, among other factors. A general rule of thumb is that the rate doubles or decreases by half for every 10-degree change in either direction. Catalyzed reactions proceed at rates vastly higher than that of uncatalyzed. We know that any reaction that is, you can say, that has a catalyst present. Catalysts are what? They increase the rate of the reaction, thereby decreasing the activation time period. So when the reaction is catalyzed, that increases the rate. Now, in the absence of any enzyme, this reaction is very slow, with about 200 molecules of bicarbonate being formed in an hour. But when we use the enzyme that is carbonic anhydrase, then 6 lakh molecules are formed every second. The enzyme has accelerated the rate by 10 million times. This is how the enzymes work. Enzymes are biocatalysts. This is the reaction wherein the carbon dioxide along with water forms the bicarbonic or the carbonic acid, and it is a reversible reaction. So this reaction, when it is done without the enzyme that is carbonic anhydrase, so it forms only 200 molecules of bicarbonate uh, in an hour, but 6 lakh molecules will be formed every second if we use carbonic anhydrase. There are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzyme, it is a metabolic path. For example, the glucose being converted into 2 molecules of pyruvic acid. What is this? When glucose gets converted into pyruvic acid, what do we call that reaction as?
Metabolic
Hmm?
metabolic pathway.
No. When the glucose turns into 2 molecules of pyruvic acid, what is that reaction? You don't remember this?
Glycolysis.
Yes, glycolysis. So why is it taking you so much time? It's the easiest question. So in glycolysis, the glucose turns, now is it aerobic or anaerobic? Hmm?
Anaerobic.
And where does it take place? The site of glycolysis is Whereas glycolysis takes place in the cell
Cytoplasm.
cytoplasm. And what kind of breakdown is it? Like the glucose gets converted into pyruvic acid in what kind of way? Like how do you define glycolysis?
Ma'am, I don't understand your question.
How do you define glycolysis? Define glycolysis.
It undergoes oxidation to form pyruvic acid.
For oxidation, oxygen will be required, isn't it? And you said that it is anaerobic. Hmm? Not studying. Hmm? So this conversion of glucose into two molecules of pyruvic acid, that is a metabolic pathway that happens through 10 different enzyme catalyzed metabolic reaction. It is the stepwise reduction of glucose into two molecules of pyruvic acid. When you study respiration, you'll know about it. Now, at this stage, you should know that this very metabolic pathway with one or two additional reaction gives rise to a variety of metabolic end products. In our skeletal muscle, under anaerobic condition, lactic acid is formed. Under normal aerobic condition, pyruvic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol, that is alcohol. Hence, in different conditions, different products are possible. Now, tell me one thing. Uh, where does aerobic respiration take place? Hmm? Where does aerobic respiration take place? The
In the
site of
cells.
aerobic respiration. Yes. Hmm?
In the cells.
I can't hear you, dear. Can you speak a little louder, please?
In the cells.
That's right. Where in the cell?
Cytoplasm.
No. Mitochondria. Hmm. How is your preparation going, Alia, for biology? Are you facing any difficulty? If you are facing any difficulty, please discuss it with me. Did you not understand the chapter?
No, ma'am, I have to revise. I haven't revised in a long time, so
Okay. All right. So up until now, everything is clear about how the chemical reactions take place, the major bio macromolecule enzymes, all of these things are clear.
yes, ma'am.
Okay. If I ask you, majority of the enzymes are what? What would you say?
Proteins.
Yes, majority of the enzymes are proteins. Now, let's just say there are some enzymes that are not proteins. So then what are they derivatives of?
Ribozymes.
Yeah, the name is ribozyme. And ribozymes are derivatives of?
Nucleic acids.
Very good, nucleic acid. All right. Moving forward, how do enzymes bring about such high rates of chemical conversion? What do they do? So to understand, we should study enzymes a little more. We have a, okay, enzyme part is there in your syllabus, right?
Yes,
Can
ma'am.
you check? It's there?
Ma'am, I'll just check one more.
Yeah, yeah, just check it once. I think it's there, but still, let's be sure.
Ma'am, here it's given enzyme types, properties, enzyme action, classification, and nomenclature of enzymes.
These things are there.
Yeah.
Okay, good. All right. Oh, okay, anyway, let's just keep it till here only for today's class because the time is not there. And in the next class, we'll start with how the enzyme acts. And there are like very few chapters left, like the nature of enzyme action, the factors affecting the enzyme's activity and everything. Okay, so up until now, do you have any doubts?
Oh, ma'am.
Okay. Now, what was I going to say? Yeah. Also, one more thing is that since after we're done with this biomolecules chapter, then we are through with the class 11 syllabus. Like we are done with it. Now, as far as class 12 syllabus goes, now you tell me what is the condition of your class 12 preparation? Like you can say honestly.
Class 12, I am fine with all chapters, but I struggle with molecular basis chapter.
Molecular basis.
Yeah.
Okay.